Good morning and welcome to OSU Extension's Soil Health webinar. Today we are going to take a look at the research and data we have on soil health in Ohio and how you can use it to improve the soil health on your farm. We're going to do our chat question this morning is what research questions do you want investigated on your farm to know how to improve your soil health? So what questions do you have about the science, the data, what the research says about improving soil health that you'd like answered through research? So our speakers this morning are Steve Culman and Elizabeth Hawkins. Elizabeth is up first. She is an assistant professor and field specialist in agronomic systems for OSU Extension. And she does some really cool work with on-farm research. And if any of you here have heard of Efields, Elizabeth is our lead on the Efields project. So I'll hand it over to you, Elizabeth.
Good morning, everyone. I'm really glad to have the opportunity to speak with you today. And, I'm going to talk a little bit about the background about the Efields program and some of the soil health research that we're starting, that we started in 2020 and we're hoping to continue in 2021. And then I'm going to hand it off to Steve, and he's going to go over in detail some of the results that we're seeing from this first year. So to begin, if you're not familiar with the Efields program, it is an on-farm research network that we've started developing here in Ohio. We began back in 2016 with our first field trials happening in the field season of 2017. We started out in just a handful of counties across the state, but as of 2020, we're now conducting trials in 39 of the 88 counties in Ohio. And you can see in this map here, all the counties that we're covering just in 2020. So it's been really exciting to watch this program grow and to engage farmers like yourselves in on-farm research with Ohio State University to help improve our recommendations and to help bring site-specific information to your operations. The thing that, if you're not involved in the program currently, that you might be most familiar with is our annual report. So this is released each January, and it highlights trials that we conducted across the state. This year's report is available currently, and you can download a digital copy at the website there, go.osu.edu/efields. If you prefer to look at a printed copy, we also have those available. You can check in with your local extension office to get one there, or you can send an email to us at the Digital Ag team and we'll send one in the mail. We just need your address and we'll get it on its way to you. So the thing that we really enjoy about the Efields program is that, it's really driven by farmer questions. All of the research that you see in that report, stemmed out from a question that someone like you had that they thought could help improve their operation. And the soil health survey that we conducted in 2020 is no different. So this time last year, we had a small group of farmers. I think I saw a few of them on the call here. They came to us and said that they had some questions about understanding soil health, how to measure it, how to benchmark it on their farm, and how to know if they're making progress. And so Steve and myself, and then a few others, sat down and put our heads together to come up with some ways that we could start answering these questions. So the research questions that we developed that Steve's going to talk about here in a moment cover everything from whether or not soil type influences soil health values. This was one when we're starting to compare fields or across farms, what kind of consistency do we expect to see in those numbers as other field characteristics change? One that was hotly debated in our farmer group was sampling depth. So there was some mixed messaging on how deep you should pull soil cores. So we wanted to understand better how much of a difference different sampling depths make when it comes to those soil health values. We were also curious about conservation practices that are being implemented and how they impact soil health. And then maybe one of these most important pieces to you on the call is, can we help you as farmers set baselines to track soil health on your farm at the field level over time to see whether or not conservation practices you're implementing are changing those values, improving them, and even more basically, what is a good value? If you run some of these soil health tests and you get them back, where do you stack up? So to answer those, the sampling protocol that we set out with was a whole field soil sampling strategy. So you can see there on the right that we're zigzagging across the field to get that aggregate sample from 10 to 15 cores. And then you can see at each individual sampling point, we were pulling 3 separate samples at those depths. So 0 to 4 inches, 0 to 6 inches, and then 0 to 8. And we kept all those separate and they were analyzed separately so that we could compare or contrast any differences we saw in the values between those depths. We set out in early spring with this project, and the bulk of these cores were pulled between May and July. To bring in that management piece, that was one of our research questions. We tried to focus on fields under 4 categories of management practices: long-term no-till, conventionally tilled fields, cover crop fields, and fields with a history of manure. And so one of the data points that we collected for each field was the number of years that they had been under these various management practices. And so Steve is going to take it from here.
Thank you, whoever is trying to find the unmute button. It disappeared when Elizabeth shared the screen. Thanks, everybody. Thanks for being here this morning. So yeah, there's a lot of questions, of course, when it comes around soil health, and we've talked in some of these at length in this webinar series. Thanks for joining this morning to talk about this. Typically, as Elizabeth just indicated, an educator, a county educator, somebody from Extension, not always, of course, sampled those fields. And then we had a total of 88 fields in 26 counties, 261 soil samples in total. Those shaded areas here in the map show where those areas came from, or those counties where those fields were in, I should say. And then, so those, soils were shipped to my lab here in Worcester, Ohio, and we ran several things. And this is what we would call kind of beginner soil health analysis. It's in no way comprehensive, but we wanted to start somewhere, okay? And one of of course, the big challenges with soil health testing is that when you're talking about adding on additional soil measurements, the price goes up, right? And so we are trying our best right now to develop tests that are rapid, inexpensive, and can be kind of rolled together so they're meaningful in different ways. They're not redundant tests, we're not measuring the same type of measurement on the same sample. So a lot more to be said about that, but you can read the slide here. We did what would be considered a standard or routine soil nutrient test that included pH, total organic matter via loss on ignition. That's a standard measurement that you get in your soil test results, and then a Malic-3 extractable nutrients. And then what would be considered new or different, the routine is, of course, what's commonly done, but what would be different, it would be these emerging soil health indicators. And we just really picked 2, and they represent other pieces of the puzzle. So the routine soil test, of course, is primarily focused on chemistry. But when we think about pH, buffer pH, and we think about extractable nutrients, what that plant is going to see over the growing season. These other 2, this POX, or permanganate oxidizable carbon, is really kind of an indicator, more of an indicator of microbial activity, biologically active organic matter, and then aggregate stability as a measure of the physical piece. So we've got really, in a very abbreviated form, we've got the chemistry represented, a little bit of the biology, and then some of the physical structure. So that's kind of some of the justification for picking these, okay? And then let me just talk, we've talked a little bit about these tests before, but for those that weren't on in earlier webinars, Epoxy or POX, permanganate oxidizable carbon, it's what is it? It's just a very simple, quick colorimetric test that really measures a small but biologically active and rapidly cycled pool of organic matter, okay? And so we recognize that Total organic matter is a very, very important measurement in soils. But if we look at some of these ones that are more cycling rapidly and active, especially within the context of soil biology, we have a better chance of detecting changes and differences earlier, and it helps us guide kind of the trajectory of where that soil is going. Are we improving soil health, decreasing soil health, et cetera, et cetera. So Epoxy as a measurement really represents a microbially processed pool of carbon. So that is how much of plant residue and other organic inputs did actually go through, in a general sense, flow through that microbial food web, and what they actually ate. Okay, and so this represents, relative. Well, we won't get into all the details, but represents. If we think about this, we believe that this is a different measurement than respiration, and respiration as that Solvita measurement might represent what's more actively available, and this is a little bit more what's processed. Both of those measurements represent kind of what we would consider a biologically active pool, represents nutrients and kind of the capacity of the storage rate, like how much actual nutrients are stored in that bank account, if you will. So we found it to be a very sensitive and rapid method, and just really is for lots of reasons is able to kind of reflect overall changes in soil health rapidly. Okay, so that's POX. And then the last piece that we measured is aggregate stability, excuse me. And just generally, this is a measurement of soil structure or of slaking. Slaking is soil when we dunk it in soil or submerge it in water, the ability of that to hold up or fall apart. So this is probably not the best photo here, but we've got an apparatus that essentially is this glorified soil dunker, if you will. There's nested sieves, and then we put soil on top, and it just kind of slowly oscillates in buckets of water. And then we find out how the ability of that soil to stick together versus fall apart, right? If it sticks together, it means that the soil is well structured, it's well aggregated. It means that we've had a lot of roots and microbial action in terms of exuding all this sticky stuff that helps bind the soil mineral component, that is the clay and silt and sand, mostly clay, but sticking that clay together with organic materials to help form aggregates. Okay, and so that's why it's an aggregate stability measurement. So there's a few different ways that we can measure this. For the purposes here, we're talking about macro aggregates. Micro aggregates form to make macro aggregates. And when we have macro aggregates, that's really where we get pore space in the soil, big kind of pockets of air that can help really improve aeration of that soil, help stimulate mineralization, soil biology. It helps with infiltration and accepting water, but we've got pore spaces there as opposed to kind of a soil that's been worked over and has very little pore space. So this is a very, very, very important measurement of soil, particularly from that physical structure perspective. All right, so let's get a little bit into the results. And the first thing is just kind of like, I think part of our experience here, we've been working on this for several years. This is really the first report from an Efields perspective, and it's very exciting to me to kind of think about integrating soil health into an Efields framework. One of the first questions is like, well, how does soil health, how does soil type, the type of soil that we are in the state, or like what you're growing or farming on in that field, how is that influenced by soil health? Okay, so what we have here is there's three measurements. There's total organic matter here, there's Epoxy, and then there's aggregate stability. It's all in relationship to CEC, cation exchange capacity. And that can just be thought of as a very kind of coarse measurement of soil type. So as CEC increases, of course, the amount of clay in our soil really does increase. So, soils in the, 15s, 18s, 20s, 25s, what we would find commonly in Northwest Ohio. Here in Worcester, it's much more common to find soils with CECs less than 10, okay? So what does this show? Well, we've got these blue lines kind of going through each one of these graphs. And we can see, not surprisingly, for something like total organic matter, which is again a common measurement that we get on a soil test, we see that organic matter increases as CEC increases typically, and that's very common. A grower in Wayne County would not expect the same organic matter levels as a grower, say, in Wood County, and that, you know, that's for good reason, because the soils there are heavier in Wood County, and they just have a natural, they are able to store more organic matter naturally, regardless if they're beat to death or not, right? They're just a different type of soil, and we should expect different things from them. The interesting thing is when you look at POX, which is again a measurement of organic matter, but again a small measurement of that, a small kind of slice of that total pool, that the slope of that line, in other words, how fast that line increases, is less with POX than organic matter. And that's because Epoxy, what we found in other studies, and it's shown here, is typically less affected by soil type than, say, a total organic matter measurement. And so again, we believe that POX is a sensitive indicator for management, and the fact that it's still affected by soil type, but to a lesser degree than organic matter, has advantages in terms of the information that we can get from it. And then finally, aggregate stability over here has a strong relationship with soil type, or CEC. And as our CEC increases, our aggregate stability, the ability of that soil just to kind of hold together and stay aggregated, typically increases. Okay, so this is all in kind of, if we take a step back, this is in a general sense, what we're really after is trying to develop or calibrate regionally within the state. If you're in, say, Northeast Ohio or Northwest Ohio or Western Ohio, what are kind of the types of values that we can think about? And so this is kind of getting at that question to some extent, and just documenting how soil type might influence some of these values. Okay, let's move on. Elizabeth mentioned one of the main things that we're after is just trying to get a handle on depth. And when we revised the tri-state recommendations over the last several years, you know, we recognized that the original tri-states were at a 0 to 8 inch depth. The new tri-states maintain that same depth, but we fully recognize that not everyone samples at an 8 inch, right? And for various reasons, you know, logistically or whatever, people might have moved to 6 inches or 7 inches or 5 inches or 4 inches, right? Depending. And one of the questions that we get a lot is like, well, should depth matter? You know, of course, for soil health, a lot of that really, where a lot of the action is in, say, that upper 2 inches or upper 4 inches, not all the way down to 8 inches, because of, you know, where that soil biology is what we're really trying to measure. So just to kind of generally address all those questions, you know, we thought about this as a nice overview in terms of looking at how depth influenced some of our soil properties. Okay, and then this is, of course, these are just averages across all those 261 soils. So there's you know, there's a lot to be said about this, but just in very kind of broad brushes we're looking at here, we can see over the course, you know, if we look at pH, there's really no difference that we detect from 0 to 4 down to 0 to 8 across those 3 different depth intervals. But if we look at other properties, if we look at organic matter, malic phosphorus, malic potassium, epoxy, and to some extent these macro and micro aggregates, this is really kind of this ag stability piece. We see in general that we have the highest values, not surprisingly, are at, you know, in that surface layer of soil. It's a 0 to 4 inch depth. And then as we move down into the 0 to 6 inch interval and then the 0 to 8 inch, those higher values in the 0 to 4 essentially get diluted down as you move through that soil and you're sampling kind of a deeper slice of that soil profile. So, you know, again, this is not really a surprising result. We fully expected to see this, but it's nice to have some data to calibrate. And just to say, like, if you are sampling at different than a 0 to 8 inch, you know, here's our average POX value if you're sampling at 8 inches. It's 583. This is a milligram per kilogram. That's basically a part per million measurement. But if you sample at a shallower depth, we can basically have on average about 100 ppm more POX than we would have, at a deeper depth. So again, this is really in the context of just trying to provide some baseline information and help us calibrate how depth might influence some of these measurements. Okay, so let's talk about management. And so this was probably the most kind of surprising or interesting or contentious finding that we found. And we need to recognize that, I guess before I've got two management slides, I'm going to talk about no-till, and then the next one will be cover crops. We need to recognize that this is not really like a controlled study where we did multiple years, some no-till side by side next to some in continuous till or whatever. This is just taking a slightly random sampling across the state and then asking growers, how long has this field been in cover crops, and how long has this field been in say cover crop management, right? And so you can see that we had some long-term no-tillers. We had people that had recently tilled. And interestingly, kind of surprising to be perfectly honest, we see that there's a slight negative trend here in terms of years of no-till and total organic matter. And the same thing with this kind of available pool of active carbon, the POX, and aggregate stability is not to the same extent. We see a positive relationship. And so we fully would expect to see positive differences or a positive relationship with ag stability and tillage. And that's kind of what we do see here. But we can see it's really noisy data, right? I mean, this is because it's not just no-till, because people are doing other things besides just a single practice. They're doing cover crops or not, they're doing manure or not, they're in different soil types. There's a lot of stuff going on here, but just if we strip all that complexity away and just look at a soil measurement and then how many years of no-till, this is the relationship that we see. So again, this is not saying that if the longer amount of time you're in no-till, you're decreasing your organic matter levels. It's not the prediction here, right? It's just showing that there's a lot of complexity. We generally, we understand, of course, that no-till typically builds soil carbon over time. It's complicated in terms of how it does that and to what extent and how fast and all this other stuff. And some of that complexity that comes out in kind of all these different studies that we have and have conducted is coming out here as well. But, this is kind of interesting. We'd like to maybe explore this a little bit more. We can get back to things we know or things validating our own worldviews. And here's cover crops. Okay, so this is the same graph, organic matter, POX, aggregate stability. And instead of years in no-till now, it's years in cover crops. And again, this is just relational. It's just a correlation. It's not saying if you're in this much, if this is not a study where we started cover cropping at different times and then controlled for all these things. It's just kind of a random observational sampling. But again, we find that there is a positive relationship with all three of these soil health indicators with cover crops. Okay, so we'd like to, moving forward, we'd like to kind of pick apart a little bit more of this and try to understand this a little bit better. This is really just, again, a very broad brush introduction in terms of the data that we have right now. Question, so just to summarize, and then I'll move on to one more thing. We're wrapping up here, folks, but to summarize, soil type matters when we're talking about soil health. This is something that we know, already exists. Soil type matters for a lot of things. But when we're thinking about soil health and needing to adjust and being realistic in terms of where we want to go with a particular measurement or a particular indicator, we need to self-calibrate ourselves for what field are we in and what's the potential of that field. And so soil type does matter. As CEC increases, as that clay content increases, total carbon and active carbon is going to increase with that as well as aggregate stability. Depth matters. We've known this, this is not new information, but depth of sampling matters. What's the quote unquote right sampling depth? I don't know the answer to that question. The answer is, I believe the answer is if you want to have it matched up with Tri-State and a lot of the work that Ohio State's doing, think about a 0 to 8 inch sample, but perhaps more importantly than that is thinking about consistency and what's the depth you have been sampling at, and then maintaining consistency over time so we can track trends, okay? Of course, we'll have some data here, hopefully that will build this up more and be able to provide some reasonable calibrations or correlations that you could say, well, I sampled at a different depth than what Ohio State does, but maybe I can actually just have a generalized relationship or convert that to what it might be from an 8, a 0 to 6, let's say a 0 to 8 sampling. So consistency with depth is just really, really important. I can't underscore that enough. And of course, management matters. That's why we're here, right? We're not here. We don't measure soil health things so we can see a value go up or down, right? So like, it's kind of like, which way are we looking at this? Are we looking at this from management to influence a soil health indicator, or a soil health indicator to reflect management? But we know that management and no-till and cover crop, those are things that should matter and should build soil health over time. And then just this, caveat, much more work is needed. This is just 88 fields may sound like a tiny bit. It might sound like a lot. It's not a little bit of work, to get this, but it's certainly not a comprehensive, state of Ohio soil health assessment. That's not what we did here, just to be clear, okay? In one of the first weeks, I promised, if I can advance my slide, I promised a little data in terms of what's quote unquote a good soil health value. This is a common question. You give a grower a new test like POX or whatever, aggregate stability, and they ask, well, what's a good soil health value? And so here's just an example of what we've done. This represents collective work over say the last 5 years in my lab, where we've run POX on a lot of different on-farm samples, and Efield data included in this, but not exclusively. So there's about 900 different soil samples here, probably from better part of, 40 counties or so, something like that. And so what we did is we just split things up by CEC. We know CEC matters. And so, thinking about lumping these in terms of sand, silt loams, and clay loam soils that we have in the state. And then we've got these percentiles, which is really just taking everything that has a CEC of, less than 6, and then ranking those, and what's in the 25th percentile in terms of the soils versus the 50th versus 75th versus the 95th. And again, we can see in this table that as your soils increase, the median, or like that 50th percentile value increases by quite a bit, going from sand to a clay loam. And so, again, what is a good value? Well, first we need to classify your field in terms of the soil type it's in. And we could break it down even farther than this, but this is just kind of a broad classification of sand, silt, and loam. And then we can look at, how your value stacks up against some of these, okay? And so if you're in a silt loam and you've got over 600 part per million POX, you're in the 95th percentile. So I'd say that you've been, it's very likely that you've been doing a lot of things to conservation measurements to improve soil health. So keep doing what you're doing. And then I'm wrapping up here, folks, but where are we headed with all this? Okay, so we would like, as a vision, but this is not a promise, but it's our vision, is just from like an agronomic crops team and Efields team, hoping to integrate soil health measurements into Efields and into Efield trials moving forward, so we can continue to capture this and just build the data that we have, okay? It's really important that we continue to support commercial testing labs to integrate soil health testing into the portfolio of tests that they offer. We've been doing some of this work, but I run a research lab, not a commercial soil testing lab. Anybody that's been a part of this knows that it took a long time to get your results back, sometimes a month or two, to get results back. It's not something that you're going to get results back in a couple of days. So let's let the commercial labs do what they do really well, which is provide quality data and really rapid turnaround time. We're continuing to work on what these indicators mean, which ones are quote unquote the best, what are good values, et cetera, et cetera. And then more importantly, maybe most importantly, is how can some of these values help provide insight to you in your operation and help guide some of your management, right? So this is like really the rub where we're not there yet. There's no question that we're not there yet, but we're trying to get there. This is the long-term goal of how does this information, right? You know how to use pH to make a management actionable decision, but how do you use POX to make a decision, right? And that's like, we're quite a ways from that, but that's really where we're trying to get to. So Elizabeth, I think I'll hand it over to you. I think I'm kind of done. I've talked plenty already. So yeah.
All right. So to wrap up here, we are planning to continue to collect some soil samples from several fields in 2021. So if you're interested in this soil health survey or any type of project that you might want to participate in with Efields, your first step would be to contact your local OSU Extension educator. You could also email us at the Digital Ag team and we can help connect you with your local Extension educator if you do not know who they are. And finally, if you just want more information in general, you can again go to that website. You can check out the report from 2020 and previous years. We do have some of our standardized protocols on the website as well. So you can get an idea of what some of these different trial projects look like before you reach out. And then the last slide we'll finish up here is if you want to keep track of what we're working on throughout the season, you can check out both the Soil Health website or the Digital Ag website. And we're pretty active on social media as well if you want to follow us on your preferred platform. I think with that, we're ready to take some questions.
Yeah, maybe I'll jump in real quick, Elizabeth. I just want to say we did have a question earlier about like, Efields, of course, is very on-farm and farmer focused. That's really kind of what we're after. We had a question about if you're a crop consultant or a co-op agronomist or retailer, how can you get involved? And by no means is it Efields exclusive to farmers only. If you're in the industry and you want to work with us or connect us with growers, we're more than happy to to help be part of that process, right? So like, don't feel like you've got to have like the grower card in your pocket to actually do something here, be part of this. And then the last thing I want to mention is just, and I'll call Bowden out if he remembers, but there is, what the data that we just, of course, presented is, just to be crystal clear about this, is in that the 2020 Efields report, right? And so it's kind of buried in the back, some page 2 something. 220, 222?
222.
222. So that's where it starts. It's like 4 pages or something. So, but go for the soil health and stay for the rest, because there's so much good information in that book. So I'll leave it at that. Please, let's move on to questions.
All right, Steve, we have a question here that I think will go back and kind of help explain a few things. Had a few people asking a little bit more about the Epoxy and what that number is saying, and for a little bit more explanation there. The question was, so say the POX number was 580, is that saying that there's 580 parts per million of carbon, or what exactly does that POX number saying?
Yeah, that's a great question. So we can dive a little bit more into that. If it's about so if you get, the commercial testing labs will report numbers a little bit different, right? So some labs or some people actually ask for the values in a pound per acre measurement. What the lab actually measures, if you're talking, let's just say something like a malic 3 phosphorus, they take an extractant, a malic extractant, they shake soil with it, they let it settle, they take some of that extractant out and they run it through and quantify, they measure what's the concentration of phosphorus in that extractant. So that's how we get a malic 3 extractant, right? So POX is very similar in that regard. But instead of phosphorus, we're actually just measuring carbon. So it's a part per million of carbon per kilogram of soil. So it's the unit is milligrams of carbon per kilogram of soil, which is just a part per million measurement. The exact same way malic 3 is measured as a part per million measurement. So some soil tests will say you got a Malic 3 value of 20 part per million or ppm. And then if you get it in pounds per acre, all that lab is typically doing is taking that number 20, multiplying it by 2 to get you a pound per acre measurement. So again, it's a measurement of concentration. If that's the question.
Great, thanks, Steve. There's another question about the Haney test, and does that have any relation to Epoxy, or do you see incorporating that into OSU's soil health program?
Yeah, it's a great question. So there's a lot to be said about the Haney test, and there's some be clear about like what that actually test is. So some people view that test as kind of the complete package. There's a few pieces to that. There's typically an HA3 extraction, which is kind of a weak Malic 3 extractant where we're measuring available nutrients or extractable nutrients, and more of a water type extractant than an actual stronger acid like a Malic. There's water extractable carbon and nitrogen, and typically total carbon nitrogen. Sometimes there's the respiration. So the Haney test is, unless they want to follow up and say specifically what they mean by that, it can be just to understand that there's like a little bit of imprecision in terms of how that's used, or it could mean a number of different things. And so we have, we all kind of come to the table with different biases, and we've looked a lot at kind of Haney values and numbers. The POX value is not terribly different than a water extractable carbon, but it is a measurement of carbon concentration in the soil. But a water extractable, or a WOEC that you get on a typical Haney test would be a much lower value because it's just really water. So we have a carbon measurement that does actually react more with carbon. It's a little bit stronger, if you will. We don't see like a super strong path in terms of merging, or like my lab, for various reasons I can explain why, we just don't run a lot of those. We feel like the measurements that we have and really focus a lot on are perhaps a little bit more appropriate for Ohio, and just they're going to be more rapid and more inexpensive, less expensive over time. And we believe that that's a really important piece to drive the adoption and use of soil health testing. So I'm not here to say that don't do Haney. That's not the message I'm saying, like don't use the Haney test, but there's a lot of options right now in terms of testing, and there's a lot of people trying to figure out what's best, what's a better test or what's a better indicator. There's a lot of chatter about that. People have opinions. Some of that's, opinion-based or science-based. Some of it, it's a complicated kind of, are you a Miller Lite drinker or a Bud Light drinker, right? It's like, I'm not going to get into that argument because it's you can't win it, right? It's like, it's hard to, so we just don't see a lot of that in my lab, but it doesn't mean that other researchers can't do it. Doesn't mean we can't send it out to have it run in addition to the stuff that we're doing as well. So, I don't know if that's a kind of a non-committal answer, but that's what I got. Mm-hmm.
All right, thanks, Steve. Going to give you just a short break here and give Elizabeth a question. The question is that samples were somewhat skewed towards Northwest Ohio. How can producers around other areas of the state get involved, submit samples as we look to continue this work?
Yeah, absolutely. We're definitely open to sampling in all areas of the state. We did have some samples pulled down in Clinton County and Fayette County, as well as in Central Ohio, but we would love to expand our data set in more areas of Ohio. So if you're interested and you have fields that you would like to be sampled, just reach out to us and we will facilitate coming out and pulling those samples and getting the analysis done. This is one of the easiest protocols on the farmer side for Efields. So definitely take advantage of the service that we can come provide.
Question about if there's any certified organic samples, Steve, and I know you've been doing some work with certified organic and soil health. So if you'd like to comment on that.
Yeah, excuse me. One of the things that we want to do moving forward is include a little bit more nuanced management information. Things like years in no-till and years in cover crop help, and those are important, like keystone things. But there's all kinds of other stuff, of course, that are going to influence these values. And just because someone, for example, might not, it might be 3 years no-till, but they might just do a light disking or something every 3 years. So it's a pretty minor disturbance. So we need to do a little bit better job quantifying that. Getting back to the management and organic management, one of my colleagues, Dr. Christine Sprunger, is, we have a manuscript that's kind of in review, and she's going to be presenting that as part of our OFA winter webinar series. Our offer winter webinar series. I do this all the time. But Ohio State has an organic program, and if I take a question, but I'll find right now and drop that into chat. But we actually part of this, not part of Efields, but part of this whole trying to get a better assessment of soil health and how organic specific management. And so these were from about 200 different or certified organic cornfields, and looking at crop rotation and perennials and hay in the rotation and use of manure and all these things. It's a really nice, pretty interesting assessment of how specific management practices build soil health in organic systems. Of course, which can be not from like the same measurements, not from an indicator perspective, but from a management perspective can be quite different than what we're dealing with in a lot of the conventional fields. So that is happening. That webinar is happening sometime in March, and somebody might beat me to it, one of the panelists on chat or something, but that I would highly encourage you to check that out, because that's going to be actually a lot more informative from kind of a how does specific management influence these values. We were a little bit limited from our study and design here, how much we could do with that, but we hope to bug growers a little bit more in 2021, annoy them with more detailed management surveys to get a little bit better sense of what was happening in these fields and what's the history of that field, so we can tease this apart a little bit better. Yep.
Thanks, Steve. On that study design side, maybe you can lead right into this next question, which was, shouldn't the organic matter graph have been change in organic matter by years in no-till, not just the number, and why we went with what we did?
Yeah, I mean, that's a good point. It's because it's not really a controlled study. We don't have a baseline, a time point 0, and then a time point, whatever. So we really couldn't have, unless I'm missing something, I don't think we really could have done that, a change in organic matter. That's like, this is the point, right? Like you'll see the no-till or the cover crop, there's a lot of sites and a lot of fields there that were time point 0, there was none. So if they're going into no-till or going into cover crop, let's go back and sample in 3 years, or let's go back and sample in 5 years, and then we can do some of that comparison. So that's really what we want to get after, and these are like what we presented here is a nice, I don't know if it's nice, but it's like an overview just to kind of get people thinking. But like, there's just so much more that we could really do with this if we had, the interests and the desire, and then of course that other annoying thing is like the funds to do it, right? We need some way to pay for these things aren't free, so we need some way or somebody to pay for this. And we're not like a commercial lab that has that whole billing mechanism set up. That's not our role, and that's not what we try to do here. So we're trying to get grant money to essentially to fund some of this work and help subsidize the cost for the growers to really help generate new information.
All right. Here, related to, there's a lot of discussion about carbon sequestration. So is this Epoxy, is it a test that'd be able to relate to possibly confirming that carbon sequestration and considering CO2 mitigation practices and different things?
We believe that POX is an absolute early indicator, very sensitive and early indicator of carbon sequestration. It's the exact same thing as organic matter storage or building rate that we believe. We've got work looking at that respiration or solvita measurement and then looking at this POX measurement. We believe that those are both important measurements. I alluded to this a little bit earlier, but for different reasons, that POX really represents how much organic matter is being stored or sequestered or stabilized in that soil. And that respiration measurement is an indicator of how much it's getting released or turned over, mineralized, decomposed for nutrient release and crop uptake. So we believe that those two indicators are very complementary in terms of what they actually reflect that's happening functionally in the soil. But POX as an indicator for soil carbon sequestration, I think is an excellent one. It's cheap and it's rapid and it's sensitive. Yep.
What types of research is OSU Soil Labs doing on biological and fungal amendments? And if they looked at the John Sue bioreactors to create seed fungal inoculants?
Well, some of that, Elizabeth can correct me if I'm wrong, I think some of that work has been done in Efields. We basically, you all can recognize if you go to any trade show or look in any trade magazine, there's a lot of different products that are on the market and more coming online, it seems, every month. We don't have the capacity to really test all those thoroughly. A lot of times they might not last very long, or they might get rebranded or reformulated. When those manufacturers or retailers, whoever's developing that product, the biological, if they come to us and ask for us to help facilitate independent trials, we often do that. There has been some of that work that's been done, but it's a moving target, and it's very difficult to really validate a lot of the products that are out there because there's so many and the turnover is so rapid. But if you're interested as a grower, get with your county educator and let's put a trial out and we'll see if we can find any differences. And these trials are typically 1 year, that's the commitment, but we can do multiple years of trials, and there are definitely multi-trial reports in Efields. So there's a lot of things that can be done.
Thanks, Steve. There's been a couple questions about where this information presented today, if we were going to share slides or if it's printed anywhere. Do you still have those slides, Bowden, on where Efields is? And if Elizabeth, do you want to answer how to get, say, a printed copy if you want that in your hands?
Yeah, I will type here the link to get the digital version of the Efields report in the chat. Everything that you saw today it starts on page 222 in the report. And if you would like a printed copy, you can send an email to the Digital Ag team. It's Digital Ag, I'll put that in the chat too. It's digitalag@osu.edu. And if you send us your address, we will mail you a copy. And then again, also your local extension office should have some available.
This will go back kind of to that discussion you've had, Steve, about the limitations of the trial design and amount of time you've had, but have you looked at splitting the study that was done for years of no-till into soil types and different soil data?
Yeah, we have not at this point. I mean, I think that we would want to collect a little bit more data before we kind of really did a deep dive into that, and that's kind of what we are hoping to be able to do, but we have not really, to be honest, done a huge amount of like that analysis that you're, like splitting things out by. The problem is that, you split things, you start breaking things out by soil type or general kind of soil type. All of a sudden your numbers go from, say 200 and something samples. Now each one has 40 or 30 or 20 samples in it. And so we just need more observations to really make that analysis meaningful. So But no, we haven't done that yet, but we want to get there. Yep. Yep.
All right, thanks. Is there a correlation or can it be extrapolated to estimate Epoxy if you know your soil organic matter and your CEC?
It's a great question. And the answer is no, not really, because, we saw, like, if I didn't show you a graph, but I could show you a graph of total organic matter and POX, okay? And not surprisingly, those things are related, and it's a positive relationship. As organic matter increases, POX increases, but there's variability around that trend line. And we believe that variability is meaningful because of how management has impacted that POX value or that total organic matter value. We know typically when one goes up, the other goes up. That's not surprising, but the kind of the relative increase in POX relative to total organic matter is an important measurement because as a soil health indicator, we believe POX is robust. That will show, for example, in Worcester, where the soil type might be, not as capable of storing total organic matter. If we can increase that POX value to get it on par with a field in Northwest Ohio, then we're doing a really good job here. So, but to answer the question, no, you can't just there's not some generalized relationship where you can just plug total and CEC in and get that.
All right. Thank you, Jason, for fielding all those questions, and thank you to Steve and Elizabeth for being willing to stay around and answer those. I think we had a lot of good discussion. Because of our time frame here, we are going to go ahead and wrap up.